Hello! Today we're going to learn some basic techniques that we use in the lab to transfer organisms from one type of media to another. And we're going to be using a set of techniques that are called aseptic techniques. And the goal of those aseptic techniques are to keep the organisms pure, to keep the cultures from getting contaminated, and hopefully to keep us from getting contaminated as well. So, the first we're going to do is a broth-to-broth culture. I have a broth that contains organisms, and I have a sterile broth. And so the goal is to take the sterile broth and transfer organisms from the tube. Anytime you use a tube, you need to vortex it, which means mix it up. Or if a vortex is not handy, you can hold it loosely by the top and flick it back and forth with your finger, and that's sufficient to agitate it. The next thing you need to do is always flame your loop. And so I have a the incinerator, and I'm going to take my loop, and you can see that the inside is orange, it is red hot. I'm going to take my loop and put it in there until the loop becomes red hot. And then I'm going to withdraw it and put it here to cool. Now, the next thing I'm going to do is I'm going to label my tube. So I have my tape. I'm going to get my tape. And I'm going to always label with the same 5 pieces of information. Your name, your lab section, that all goes together, name and lab section, the date, the type of media, this is nutrient broth, so I'm going to write NB, the organism that we're using, this is Chromobacterium violaceum, and your temperature. This is going to be grown at 37 degrees Celsius. So I'm going to go ahead and I'm going to put that on my test tube. Now a lot of people will wait until they're done doing the culture to label it, and then they have multiple test tubes and they forget which is which. So I would always encourage you to label it first. So I've got my sterile loop, I've got my tube with the bacteria, and it's been mixed up. Whenever you remove the lid, I'm going to be holding my loop, and you hold it like a pencil. Take the lid off with your pinky finger, and you're going to insert the loop. You want to move the tube, not the loop. And so you're going to move the tube, dip the loop into the culture, and put the lid back on. And now I'm going to do the same sort of technique to put this loop into my pure bacteria culture, which right now is sterile. Take the lid off, hold it between my pinky, keep the loop still, move the tube, give it a gentle swirl, and now I can retract and put the lid back on. The reason you don't want to move the loop is because there's bacteria on it, and if you go moving it around, then you can spread the bacteria everywhere. Now before you use this loop again, you have to sterilize it. So again, I'm going to flame it. So I'm going to put it in, hold it there until it's red hot, and then withdraw and place it to cool. Now the next we're going to do is a slant. And so here we have a slant with bacteria. And you can see it's called a slant because it's agar in a test tube, but it's been cooled at a slant. We're going to do a slant to slant culture. Because it's a solid, we don't need to vortex it, but we do need to label. So I'm going to go ahead and label my tube. Again, my name and lab section, the date, the sample, which is Chromobacterium violaceum, the type of media, nutrient broth, and 37 degrees Celsius. And I'm going to go ahead and put that on my slant. Now my loop is sterilized because I've already done that. So I'm going to take my sterile loop and I'm going to take my slant. Remember, I take the lid off with my pinky fingers of the loop hand, and I'm going to move the tube, not my loop. So I'm going to take the loop and gently touch it on the surface of the media. You don't want to take a big scrape, you just want to touch enough and pull it out. Notice I'm moving the tube and not the loop. And now I have bacteria on my loop. I'm going to recap the tube. Going to place the tube in the rack, and now I have my tube with the sterile slant. And I have my loop with the bacteria on it. I'm going to remove the cap. And I'm going to hold the tube with the auger up. I'm going to move the tube so that my loop is gently touching the auger, and I'm going to fishtail it back and forth on the auger. And then I'm going to retract the auger, cap it up, and I will flame my loop. Hold it in until it gets red hot, withdraw, and place to cool. The next transfer technique is going to go from a broth to a sterile slant. Again, you vortex up your broth, or for this case, we'll just flick it back and forth because there's so little in the tube. My loop is already sterile. Always label your slant. So we're going to label the slant. With the same things: name, lab section, the date, the type of media, nutrient broth, 37 degrees Celsius, and this is Chromobacterium violaceum. Notice that the order that you put the information doesn't matter. What's important is that you have the information. So I'm going to take my tape and put it on my sterile slant. My loop has been sterilized. My broth has been vortexed or mixed up. So I'm going to take the broth, remove the lid with my pinky, take the loop, don't move the loop, move the tube, and I'm going to move the tube so I get a sample, and I'm going to retract it. Notice when you retract, you want to be careful not to bang the loop against the lid. Now, I'm going to take my loop and take my sterile slant. I'm going to remove the lid with my pinky. I'm going to move the test tube, hold the test tube with the auger facing up. And I'm going to do my fishtail, the highly effective squiggle. Withdraw, cap, return this to the test tube rack, and, as always at the end, flame your loop. Put it in until it gets red hot. And notice it doesn't take that long. And then withdraw and allow to cool. The next thing we're going to do is how to go from a slant to a broth. So we have our slant with the bacteria on it. We have our sterile loop that's been sterilized in the flame, and we have our broth. As always, we need to label our test tube. So we're going to go ahead, get our tape, our name, the lab section, this is going to be Chromobacterium violaceum, and here I'm going to write from slant, just to distinguish it. 37 degrees Celsius, and this is nutrient broth. We'll take the label and we'll put it on our nutrient broth. And now we're ready to go from a slant to a tube. So we're going to do the same thing. I'm going to take my sterile loop, going to take my slant, I'll remove the lid with the fingers of the hand holding the loop. I'm going to take my loop, put it in without touching the walls of the tube, gently touch it to the bacteria, and withdraw it. Trying not to catch the tip of the loop on the cap of the tube or the neck of the tube. Now I have my sample on my loop. I'm going to take my broth, going to remove the cap with my pinky, going to move the tube, not the loop, and now that's in there, I'm going to give it a good swirl. I'm going to retract it, and I'm going to tap off the extra liquid, and I'm going to recap. And then last, I'm going to flame my loop. Put it in until it gets red hot. Withdraw and allow to cool. The final transfer we're going to be doing is from a plate to a broth. So this is my plate, Chromobacterium violaceum, and I have sterile broth. As always, I'm going to label my tube. So I have the name, my lab section. This is going to be nutrient broth. This is Chromobacterium violaceum. From plate. And I'm going to be growing this at 37 degrees Celsius, and you need the date. I'll take the tape and I'll put it on my sterile broth. Now I have my sterile needle, and I'll take my plate. We always store plates inverted, so I'm going to go ahead and flip the plate over and remove the lid, but I'm going to use the lid as a shield. I'm going to take my loop and touch it to a colony. Now I have my sample on the loop. I'm going to take my sterile tube, remove the cap, move the tube to my loop, give it a good swirl, withdraw, tap off any extra, and replace the lid. And then finally, I'm going to sterilize my loop one last time to kill any organisms that are on it. So leave it in there until it's red hot. Withdraw. Because it touched that, I'm going to go ahead and do it again. If you bang your loop up against anything or accidentally touch it against anything, go ahead and re-sterilize it. It takes a couple seconds to do. Nice and red hot. And allow it to cool. And so those are the ways that we'll be using to transfer bacteria from one type of media to another, called aseptic techniques. Next lab, we'll go ahead and look at the results of these. Thank you for watching.